right, well, I would say, it looks like a pretty good group. We could probably go ahead and get started. And so we are today. Thank you all for joining us. This is the first of the Northeast IPM. The question series, no longer stupid questions, now smart questions. So this is the first one of the year. We have 4 total. We'll talk about that a little bit more at the end. As a reminder for anybody who hasn't been here last year, it's a pretty informal session in general. So we have 2 speakers today talking about bitter rot, biology, and management. But it is, again, fairly informal. So if you have questions, you can feel free to jump in at any point or save them. We'll maybe take like a little break between the 2 presentations just to really open it up for discussion and questions. So our first speaker today will be Philip Martin, who currently works with LabServices Life Science as a tech support agent. But many of you guys will know him as the former PhD student with Carrie Peters studying bitter rot. And that's what he will be talking about today. So you can go ahead and share your screen and take it away.
Unmute myself and get this to work. Okay, can you see my screen?
It looks great.
All right, so I just titled my my presentation here about the biology of bitter rot and some of the history of management. So hopefully you don't see apples like on the back of this opening screen here, but this was in my PhD work at the Fruit Research Center, and we certainly got some infections. Well, okay, that didn't go as planned. Am I at the wrong end of my? Okay, that's better. Yeah, so just a little bit of background on me. I know many of you know me, but yeah, I was born and raised on a small farm in southeastern Pennsylvania. I went to college a little bit later in life in my upper 20s, and then I did my PhD under Dr. Carrie Peter, as Janet mentioned. And there's some shots. I'm sure these look familiar to Mike. But yeah, the Fruit Research Center and the graduate housing. It's a beautiful place, and it was a great place to do my PhD work, which I did on bitter rot of apples. So I'm drawing, mostly from that research in my presentation today. And yeah, currently at LabServices, and we do contract research for companies doing mostly efficacy field trials. So just identifying bitter rot, I'm sure most of you are quite familiar with it, but it's identified by the sunken lesions. It has a V-shaped lesion in the apple flesh, and often, but not always, it has these orange spores, these conidia, and concentric rings of acervuli, that the spore producing structures. It's caused by fungi in the Calototricum occultatum and Calototricum gliosporoides species complexes. So there's a number of species that can cause it. There's a few most common ones, but these 2 species complexes, Calototricum species complexes. And I'll go into the briefly go into the disease cycle. So you have your overwintering structures. I think those are often mummies in the fruit. Mummies in the tree canopy. They can be twigs, possibly buds, but it's largely it's plant material. They're not soil borne. It's infected plant material. And overwinter produce spores. They land on the fruit surfaces where the spore produces that aprosoria, where it punctures through the apple skin, and first it produces these endophytic or latent infections. So you won't see the rot initially. You might see I've seen a tiny red spots that are just indicative of injury, but these can be latent or just sort of dormant for quite a while after that initial penetration of the apple skin, and you might not see the infection till post-harvest. And not in my research, but other people have shown they can actually produce some spores in this stage. Not many compared to later on, but just a little bit. And then of course you have your necrotrophic infection where you see the rot and masses of orange spores and the classic bitter rot. So I have this general disease cycle. See, I think I'm missing an arrow here, but it's polycyclic. You can go through your overwintering, but as soon as spores are produced, they can reinfect. They can reinfect from necrotrophic infections, and it can just cycle, throughout the season. Now, the ideal conditions are warm and wet. So usually we don't, we assume a lot of these infections aren't happening until June or July. I mean, they could happen earlier. I think they do, especially down south where they have warmer conditions. So back to the fungal species that cause bitter rot. So we did a part of my PhD, and other people, surgeons, worked at this and in collaboration with Srdjan, but we looked at what are the most common species. And this one here in red, Calototricum fiorinae, was the most common in Pennsylvania and surrounding regions. So with my PhD research, we looked at this one a little bit closer with its, you might say, its ecology, because there were other papers that mentioned that you can find Calototricum fiorinae in leaves in many broadleaf plants. So I started looking, and I collected leaves of various shrubs, wild grape, amber honeysuckle, poison ivy, so on, and we would surface disinfest them to take care of the microbes on the surface. And then I'd put them in a freezer, and then to bring them out of the freezer and just incubate it. So the idea with the freezer, that would kill the leaf, but it wouldn't kill the fungus. And then afterwards, the fungus could just take off if it was present inside of the leaf. And we found, a lot of these leaves, not all of them, but a lot of them just sort of erupted with these orange spore masses. So, Calototricum fiorinae really infects the leaves of a lot of broadleaf plants. Now, it doesn't cause disease in those plants, but it's there. It's there as an endophyte, just underneath the skin, just happily hanging out there. And then we took this research a bit further, and I collected leaves from broadleaf trees in a woodlot adjacent to the Fruit Research and Extension Center in Biglerville, at this bottom arrow, and also from the Michaud State Forest, about 12 miles to the northwest here. The idea that Michaud State Forest, there's no ag for at least 4 miles in any direction, as opposed to the woodlot adjacent to our orchard. And we found leaves with Calototricum fiorinae in both places, quite abundant in both places. So we concluded that, at least for Calototricum fiorinae, it really is primarily a leaf endophyte. And we came up with this infection cycle for Calototricum fiorinae. Now notice that this is not for bitter rot; it's for the fungus. So we, our hypothesis, or I think the evidence points that naturally this fungus just lives in the forest and broadleaf plants, and it infects the leaf, and it produces some spores from that leaf infection, but it never produces necrosis. It's never, you don't see any disease, and it can just cycle through the forest or through your fence row canopies, just throughout the year. And then if there's, only if there's susceptible fruit available will it cause these necrotic infections on apples or on other fruits like strawberries and blueberries and so on. Now back to the bitter rot disease cycle. This is say Calototricum fiorinae or the other species on apple, where it's just cycling within say an apple tree canopy. But one thing we did check, it can infect apple leaves as well. We found it in apple leaves, and again, no disease on the apple leaf, just as an endophytic infection. So a little bit on the history of bitter rot apple management. This was something I looked at, sort of, yeah, moving from biology to management. But looking at the history, when I did my graduate work, I spent a bit of time searching through this Haithi Trust website. It's a lot of scanned documents, and I would look for bitter rot. And it was just interesting looking at the history of, you know, what people wrote about bitter rot over the years. So hopefully this isn't too boring for you, but there's no tests, so this should be a light listening. But back as far back as 1803, there was a domestic encyclopedia and a few other orcharding documents that listed this Van der Veere, if I'm saying that right, this one apple cultivar as being highly susceptible to bitter rot. I don't know if these publications were all referencing each other or coming from the same source, but the key is, they noted that there's some cultivars are much more susceptible than other cultivars. That's kind of the point here. There's this doctor who was actually looking at I think influenza in Rockingham County, Virginia, and he had this very folksy description of here's the area of Rockingham County, and here's the diseases that the people have. Then he also, in kind of a side note, he noted that apples in many orchards are rendered unfit for use by what is called bitter rot. And he mentioned a disease of which the cause, so far as I have been able to learn, has not been well ascertained. And so this was 1807. So you see, this was a problem. I mean, as basically as far back as we have records. And it makes sense if the fungus is native to our forests, then it makes sense that the fungus was always there. It's just a matter of having the susceptible plant in the right conditions. In 1842, there was a subscriber from Southern Ohio and this Western farmer and gardener who wanted to know about bitter rot, from whence it came and when and where it originated, and above all, the cause of and prevention for the disease. But anyway, this same subscriber had some opinions on what might be making it worse. And trees that are crowded suffer more than those that are well exposed. So your leaf wetness or your fruit wetness hours, and the ones on the hills escape better than those in the river bottoms. Again, the wetness. But the point that I found interesting is he mentioned that the people of this and our neighboring states have worse than thrown away thousands of dollars within a few years past by employing men from the north to head down and engraft their orchards, who make practice of fetching cuttings from the north and giving them a great name, but not at all adapted to our climate. And I think that was pretty perceptive there. Again, you have your cultivars that some cultivars are much more susceptible than others, and cultivars that might do well up north where you have cooler weather just are very susceptible to bitter rot further south. This was a theme that kind of kept going with the late 1800s. You had the USDA start up and a lot of experimental stations and a lot more publications on bitter rot. But in 1894, this William Allwood from Virginia noted that it's bitter rot's distributed across the U.S., but not nearly as serious in the general area as it is throughout the region, or not nearly so serious north of Maryland and the Ohio River as it is in the southern and southwestern states. And he talked about this strip, Virginia, West Virginia, southern Ohio, southern Illinois into Arkansas, that has, a lot of bitter rot. And again, I think it speaks to the warm, wet climate. In my own research, I just did a survey of growers asking them, by cultivar, if they could estimate how much they lost to bitter rot. This was in 2018, which was an exceptionally wet year. It was like a record-breaking wet year for us with lots of bitter rot. And the key is, it's kind of hard to read on this graph, but the black bar is how much they estimated they lost to bitter rot. And the point is that it varies a lot. You have some down here, Evercrisp, Jonathan. I mean, not a lot of, Stamen, 10 growers reported on that. Compared to, say, Northern Spy, Nittany, Empire, Honeycrisp, where growers are reporting quite high losses. And note here the range of losses at the right, it varies a lot from grower to grower. Some growers have high losses, some growers don't. And I'm sure you've all noticed this as well, but Empire growers reported 0 to 100% loss. This in 2018. But I think, a theme, started back from the publication in the early 1800s right up to my own, cultivars vary quite a bit from one to the other into their susceptibility to bitter rot. And I just a note, this is actually a cold hardiness map, but the idea is that, differences in temperature, you look at where Honeycrisp originated, Empire originated, and at least in Pennsylvania, where we're trying to grow apples. And it's kind of like that, subscriber from Ohio noting that, these cultivars from up north are trying to be planted further south, and we're having a lot of trouble with bitter rot. So moving into chemical management, chemical, I mean, the historical publications talk about chemical management from the early 1900s on. And in this one, let me move my little screen here. But this is from the proceedings of the Cumberland Shenandoah Fruit Workers Conference back in 1938. And this was a trial out of Delaware where they were looking at bitter rot, controlling it with Bordeaux mixture. So your lime copper combination. And if you look at the right there circled, they were doing quite well. Their untreated control had 99% bitter rot, and they were down at 14%. Excuse me, 14% with their Bordeaux mixture. But the key here is, if you note the next column, they have copper fruit injury. No copper fruit injury on the untreated check. Around 40% of the fruit were injured by copper in the Bordeaux treatment. So this was a common theme in publications from 20s, 30s, 40s into the 50s. They had products that could control it reasonably well, but these products were corrosive and caused a lot of phytotoxicity. And then just moving, this is fast forwarding super fast, but products like Captan and EBCDs came on the market, did fairly well with bitter rot. In my own research, I did a bunch of lab trials and also field trials, and I came up with this graph of on the right as some of the listed from top to bottom as some of the better active ingredients at controlling bitter rot. But still, the multi-site mode of action fungicides like Captan are still really a lot of the best fungicide options. As far as what's currently good, I'm gonna push most of that off to Srdjan. He can take it from there. One thing I'll note with looking at peer-reviewed papers and fungicide efficacy, what works well in the lab does not always work well in the field. You'll notice in my graph, there's these gray lines, which is frac group 3. Something like difenconazole looks pretty good in the lab. This is the results of my EC25 values in the lab. But in the field, it just doesn't do as well. And the whole frac group 3 just doesn't do as well in the field for reasons I'm not quite sure of. I know I think Guido Schnabel from Clemson did some work at the genes involved in that. But so just a word of caution. If you see a publication showing it works excellent in the lab, it may or may not be excellent in the field. Back to the bitter rot disease cycle. Thinking of bitter rot management. So if this is the cycle, I did a bit of thinking in my PhD as to what percent of the spores or the effective material makes it to these next steps. And I changed the graph to something like this, where I identified about 4 key transition points. So one, you have your overwintering structure, and you have the spores produced from that. The majority of spores that are produced from these overwintering structures don't land on the fruit. The fruit are small, they don't land. So they're dispersed. They might land on fruit surfaces or on leaves, get infected. But of all the spores that land on the fruit surface, only a very tiny percentage of the spores that even land on the fruit surface cause infection. The wetness has to be long enough, probably more than 12 hours with the right temperature. Probably, lack of UV light, all the other right conditions. And especially with the spores land on fruit surface, the step 2, non-viable spores. I think this is where our fungicides are most crucial. We put the fungicide on the fruit surface, the spores land there. Even if they start to grow a little bit, the fungicide slows them down enough that, in a day or two, the sun comes out, it dries out. The spores never get to the point where they penetrate the apple surface. And the length of the 2 arrows is here as to about my estimation as to basically what portion of the spores would make it to the next step. I think just a very small percentage of the spores even land on the apples, and a very small percentage of those create disease. And anything we can do to just make that number even less would help us with our management. And then once you have the infection in the apple, the switch to necrotrophy. I had some data that indicates that a fair amount of them, it could be half of them, it could be, but a fair amount of the ones that cause these initial infections will become necrotrophic, assuming the apple becomes fully mature and there's no post-harvest fungicide treatment. There is, as far as post-harvest fungicide treatments, at which point the infection would already be there, fludioxonil, or the Scholar, seems to be really the only one that has a evidence that it reduces bitter rot, and a lot of that was that was back 15 plus years ago. Dave Rosenberger did a bunch of post-harvest studies, so that's that source of information. Fludioxonil also looked really good in my lab tests. But for the most part, I think once the infection is in the apple, there's not a whole lot you can do to keep it from switching to necrotrophy. And then, of course, survival to the next season. So removing the mummies from the tree, just standard good horticultural practice. Removing dead branches, removing fruit mummies. One note with this, we have necrotrophic infections. I did a small experiment in which I took an apple, bitter rot infected apple, necrotrophic, producing spores, and I tried to quantify how many spores one infected apple made, and it was in the billions. I forget the exact numbers, but it was billions of spores. And so if you think of one infected apple, could infect, say, a few more apples. It could infect dozens of more apples. But if you think of the fact that, one infected apple producing billions of spores that might infect, a dozen more apples, the spores that are produced really only have maybe roughly a 1 in a billion chance of actually producing another infection. So the odds are kind of stacked against the fungus, but it just produces so many spores that they still get out, they still cover. And if you have susceptible cultivars, you definitely still get infections. Susceptible cultivars, warm, wet conditions. And so with that, I just put my opening slide back around at the back end as well. And so that's very fast, very brief on some of the biology and history of management. And hopefully that would stimulate some questions or give a good springboard for Srdjan to take it from there.
Yeah, definitely. That was great. Thank you for all of that. I really enjoyed, it's not often that we hear that sort of like history of the disease coming in and how the sort of cultural history of people moving apples around and the disease changing and morphing, and then also the management. So that was, I thought that was super interesting.
Okay, well, that's good, because I was debating how much of that I want to put in. I mean, there's like one thing I didn't put in is, there's one recommendation from, I forget, the, end of 1800s, sort of pre-USDA, was like, run hogs in your orchard so they can clean up the rotten apples, and it was some interesting things it makes sense, but have maybe limited value for our commercial growers.
Right. Makes a ton of sense. Yeah, it's, well, that's a whole nother conversation, but yeah, too bad that that's no longer commercially necessarily viable. So we are, we're doing good on time. Maybe we'll just take a quick pause here. And if anybody has any specific questions about the biology or the history of the management before we jump into Srdjan's talk.
Well, I have a question I can ask. It's probably a dumb question. So going back to our original days here, um, do you have a sense, Philip, where the dividing line is? I mean, as far as how warm it needs to be for this to be a big problem? Because I feel like for
Mhm.
me, the guys I work with near Albany, it tends to be an issue, but then I don't see as much of it up in the Champlain Valley. So
Mhm. Mhm.
maybe we can kind of crowdsource this. Do we have a sense of where that dividing line is in as far as how north we need to be for it to not be an issue on our hunting crisp?
Actually, would like to interject here a little bit and just to remind you that I've heard a story from Dave Rosenberger. That actually, Forenses had issues with bitter rot in the past. Okay, and for several years they have been battling it, but they realized actually Dave realized that the orchard that had issues with bitter rot up there was very close to a woodlot. And at a certain point, the Forenses he just told them just cut that woodlot, and it was a very small, like a meadow woodlot, section, and they cut it out and pulled it. I would agree with you that because of the latitude and the temperature, I assume, or maybe some other conditions that we are not aware, it's not that terrible. But as you go south more to the southern part of the Hudson Valley, and as you go south to Pennsylvania and then Virginia, it becomes much more devastating because the conditions during the summer are the ones that really drive the devastation, I would say.
Yeah. Yeah, as far as if you're looking for a temperature number, at least in our lab tests, the low 80s, 25 degrees Celsius, that's about the optimum for these fungi. But the interest, and I pondered this in my research because it seems in the field, you don't really get bitter rot until you get temperatures maybe, reaching 90 or in the 90s with warm weather. And so in the lab, if you're trying to grow the fungus at 90 degrees, it's actually starting to slow down already. The fungus is stressed. And so I've wondered if, 90 degrees is also stressing your apple. And so maybe it stresses the apple more than it stresses the fungus. And so the fungus still gains the advantage at that, 90 plus degrees. The other thing is, if you hit a high of mid 80s in the day, but you're still cooling down to, 70 degrees at night, you're thinking of the wetness period. The wetness period might be overnight with the dew or, a rain. So I think, if you're in the mid 80s, average in a 24-hour period, that it makes sense. Then you have your optimal temperature for a long period. And again, the hot temperature, yeah, the only thing I can think of is it stresses the apple more than it stresses the fungus, and then the fungus is just poised to take off. And also, it seems to me that you can create your disease prediction model where you just put in temperature and you put in leaf wetness, and it works reasonably well. But I do think there's something about inoculum buildup in an orchard, because you can have the ideal conditions in I know up there in Champlain Valley and places, you might have a few days in the summer where you get warm and wet just for a brief period of time. Or I mean, correct me if I'm wrong there, but you can get these warm conditions, but it seems um, you get the same conditions further south in an orchard in which bitter rot's been a problem for years, and you get more bitter rot than the same conditions in an orchard where it hasn't been a problem. I guess the other background there is even here in Pennsylvania, some orchards struggle. Some orchards, not that far apart, have one orchard might have a lot more bitter rot than the next orchard, and the differences in temperature are not that dramatic. So I think there's several things going on, and I'm guessing the other one is just the inoculum pressure. But anyway, I'm rambling a bit. I think it's 80 degrees and higher is optimal.
All right, sounds good. Thank you. We're definitely more and more increasingly hitting those, so
Mhm.
definitely keeping an eye out. And that's good to know about that orchard you mentioned, Srdjan. I'll have to ask them a little bit more about that.
Or
Thanks.
follow up with Dave. He can tell you definitely a lot of stories.
Oh, sure. Thanks.
I've got one quick question, if it's okay. So great presentation, Phil. Thank you very much. I just wanted to ask quickly, we have a grower here in New Hampshire, and he is kind of early on in the establishment of his orchard. I think the trees are now 3 years old. And he's doing a multi-leader system under cover. So he's kind of got rain flies in place, and he will employ some netting on the sides for insect exclusion and that sort of thing. But it sounds like from what you were just saying and what I know about this disease, that should be a fairly effective strategy for bitter rot control, just excluding rainfall, right?
Yeah.
And keeping those leaves dry.
Yes, definitely. And that's, I just assumed, everybody's here in the Northeast with warm, wet, with wet conditions. But, if you look at the, say a Washington State publication of apple diseases, oftentimes bitter rot's not even on the list. I mean, it's just too dry. And there was a real interesting, short paragraph publication coming out of Israel from, I think the 1970s, where they were growing it again in dry conditions, but they had overhead sprinklers. And they talked about, hey, they started getting problems with bitter rot. They moved the irrigation from overhead to under tree, problem eliminated. So,
Okay.
yes, yes, I would say if we can, yeah, warm, wet. I mean, anywhere you can sort of cut that disease triangle, I would anticipate, and unless there's a lot of dew or some other source.
It's definitely probably true for the, for certain locations. But when you come down to North Carolina, actually, Sarah Villani made a presentation, really good presentation, at the Hershey last year, where she basically said that us orienting the application of the fungicides based on the amount of rain that we receive, she thinks that it doesn't work. Like 2 inches of rain as a cutoff point doesn't work for her anymore. And she thinks the reason why is because of the dew as a factor. That is another thing that they have. So they have dew up until noon in the mountains in North Carolina. So the leaf wetness would be definitely probably a good indicator of it. But if the growers spray every 14 days or 2 inches of rain, she thinks that rule falls apart where she is because of the dew.
Yeah, that makes sense. So there's always the question, how much dew is enough dew and for how long? Yeah, but we do have good, we have a lot of weather stations out, in the Northeast, and we're monitoring leaf wetness for sure. So that should give us some good information about, at least, help us make decisions about when it's time to go back in. Great. Thank you both. Yeah, I'll keep you updated on that. If we start seeing bitter rot in that system, which I don't anticipate, I'll let folks know, but should be interesting to track.
Sure. Yeah, and I think there was actually work done on anthracnose of strawberries, which is the same fungi, by a guy from Ohio State, the epidemiologist, Larry Madden, back in the 90s, looking at temperature, because he could grow these strawberries in the growth chamber, and there the optimal infection temperature was higher than the optimal temperature for the fungus. I think that's the other background to my previous statement about temperature. So yeah, I'm just throwing it out here with I have time. Another thing that I pondered in my PhD research is if there's any correlation, not really, it could be causation, but certainly there is a correlation between some bitter pit and bitter rot. So if Honeycrisp gets, notorious for bitter pit, and it also gets bitter rot. And just speculation here, hypothesis, but if you think of bitter pit and the xylem vessels are breaking down and the tissue is starting to break down from bitter pit, what it seems that would make just apple defenses are probably also breaking down, and I'm wondering if there's some connection there, so anyway, just that's me hypothesizing, but yeah.
Are there any other questions on the biology? This is, I don't even know if this is a question specifically. It might also be an attempt at speculating out loud, or maybe it's just another stupid question. But as an entomologist, I feel like the way that we think about the landscape level pests that are really present in the woodlots surrounding the orchards is very different from the way that we might think about a pest that's a little bit more apple specific and that's more kind of confined into the orchard. So my ears kind of perked up when you were talking about just finding it out there, spores on nearly every leaf in the woods. So I'm wondering if that is also true with diseases and if there's a way that that can help to kind of frame growers' perspective on thinking about bitter rot, or is that not really relevant in this situation?
I think it is. I think, one, you can't eliminate it. It's there, but I think concentration of spores makes a big difference. Even if you have only, 1 in a billion spores actually successfully creating bitter rot, if you have trillions of spores out there, you can still have thousands of apples being infected. So, still doing all your good horticultural practices to keep the spore load down, just knowing you're never gonna, you're not gonna eliminate it. One of the things that I in my PhD work, I gave the growers a survey, and one of the questions was how it presents itself in the orchard, because this is back in, old data. Some of it talked about certain trees being worse than certain other trees, and they were talking, the big 30-foot full-size trees. But I asked growers if it's uniform, do they have hot spots, is there a gradient? And the answers were all, it's either uniform or then there were certain hot spots. But I anticipated if there's a gradient, say from the woods towards, farther away from the woods, more people would observe that. Well, I think only one grower checked that box, but I think he had checked all 3 boxes. So basically I concluded that growers and just me talking to growers, asking them how does it present in their fields, they're not seeing, nice gradients. And if there is a gradient, it tends to be more, bottom of the hill, top of the hill. I've heard that, just more leaf wetness, almost more than inoculum. So I don't know if that's because the trees are just far enough away. They're rain splashed. So it's rain splashed. Mostly you're talking about feet, not yards, except in storms where it could go for a long ways. But there's not a I think it helps to frame, the fungus and that it's always going to be there, but there's no direct, do this to your wood lot and then therefore this will happen to your orchard. It probably won't make a difference, but as Dave, Srdjan mentioned, Dave Rosenberger saying it might, depending on your situation, but not consistently enough that we can say this practice will give you this result.
Yeah, that's actually really interesting that there hasn't been an observed gradient, like you were saying, like you might expect. So
I think there's just enough of it in the orchard that, even if it's coming from the forest on the edge of the orchard, there's enough of it coming from within the orchard on the other end that you don't really notice the gradient.
Any other questions specific for Philip, or maybe I guess let's push ahead and then we can always, any of the questions at the end, we can loop back to either of you. So our next speaker.
Can I make a quick
of
stupid question, maybe a stupid comment? Just all this discussion of temperature and moisture conditions at the top and bottom of the hill in different places. I was just wondering if this is an opportunity, or maybe this already exists and I don't know, like a table of wetness and temperature that correlates to an infection, like the equivalent of a Mills table. Maybe there should be a Martin table. Have you thought about that?
Well, actually, there are models that are out there. Okay, and I mean, I'm going to let Philip, give his 2 cents. One of the things that I have done is I have found the Excel version of the model from South America, and it is from Brazil. Okay, and we will be doing some work trying to validate whether this model works fine, but at least for now, it has been implemented in Catalonia in Spain. And they have digitized it and the growers are using it. It's a new disease there. And so it has, the way how it functions is the leaf wetness and temperature, of course. And one thing that the model produces is the percentage of infection you should expect with a number of leaf wetness hours at certain temperature, or the flux of the temperature, how it changes. So, and it can also give prediction of the infections on the leaves and on the fruit. So we would love to also, and I've met with Carrie Everett from New Zealand. She also developed a model for Qualitatum Furinia. So we're going to try to do some work, probably also collaborate with New Jersey, with Caitlin, to try to see if we can actually have 3 locations and also work with Scott, try to see if we can validate these models and see which one works better before we can actually recommend something.
Yeah, I mean, I would say it's hard to do good studies in apples because they're so big, and your growth chamber studies. So I think the best approximation is going with the strawberry model. I know Scott did a bunch of work with his grapes, and again, some of the same pathogens. And one of the questions for me is just is there, how close is the strawberry model and the grape model to, say, an apple model? And had I continued in academia working with bitter rot, that's definitely something I would have pursued, is sort of trying to field validate, these models for, some that are apples, like Srdjan mentioned, New Zealand and so on. A lot of them, they're very similar. I mean, I've played around with them. Yeah, you more wetness, more temperature. They use different equations, but I think there is the part of the problem, though. The real big problem with the model is it tells you when to spray. Like, it tells you when you should have sprayed after the fact, because the best control is preventative spraying. Get some cover on there so that when it rains, the apples are covered. Well, you have to go by the weather prediction, and especially in the summer when it's warm and wet, most of that precipitation is thunder showers, and they're super unpredictable. You might get hammered, and your neighbor 10 miles down the road barely gets a drop. So it's hard to use the weather forecast to tell you when you should be spraying. It's easy, even if the model works, it'll tell you afterwards that, yeah, you should have sprayed 5 days ago. I don't know. I mean, so there's some of the things I've thought about with using models.
I just wanted to hop in and just mention that I did do a trial this past season. I incorporated that strawberry model you mentioned, as well as the South American one. And Srdjan and I are planning to work on this more. But basically what I've found, and to answer your question about whether they really line up, is they pretty much all line up. So the days where one model said that there was going to be infection, all the other models also said that there was going to be an infection.
Oh, great.
For the most part. And it really depends on, you have to set like a threshold for each of these models, because they give you a predicted, percentage of fruit that will be infected. You have to set a certain threshold, where above that threshold, that's when you are going to spray. And pretty much they all tend to predict the same thing. So kind of interesting. But I was also relying on kickback activity, kind of like what you're mentioning, Philip. So I was measuring, had leaf wetness sensors, temperature sensors in the field, and I was spraying, after these infection events happened, as soon as I could. And I think if you select the right fungicide, it might work. I was sticking with Captan just so that I was able to spray it as many times as I wanted. And I could keep it consistent between all the treatments. But in the future, it might work better with a different mode of action that has more kickback activity. And I also want to try it with forecasting as well, just how well that works. So,
yeah, well, that's exciting. Glad you're taking that. I mean, that's good information.
We hope to get money for it.
yeah.
That seems like a really good segue to move forwards into the next section, talking about some of the management. So thanks, Philip, for that really good presentation, and everybody for your questions. Our next half of this presentation is going to be Srdjan, who will be Srdjan is the tree fruit pathologist at Virginia Tech, and is a very familiar face to everyone in the group. So I don't need to introduce him too much, but he'll be talking about sort of management programs for bitter rot.
Thank you, Jeanette. So yeah, this is not only my work, although a lot of it is done by me. This is my team, and some of them you met as well. So I'll just go right into it. Philip mentioned the cycle, and just as a reminder, this is how these reproductive structures look like. On the left side, you can see perithecia, which are these pear fruit-looking bodies. And I think that's what Sarah in North Carolina is dealing with because of the conditions that are probably more likely conducive to produce the perfect stage or the sexual stage. So these spores are airborne, the sexual spores, whereas the ones that Philip mentioned that are produced in the acervuli. So these orange droplets that you see on the fruit, they are basically originating from these acervulites, basically plate-like looking bodies that produce a bunch of conidia on the conidiophores, and they're rain splashed. So you can have multiple cycles of these spores. That means that, as Philip mentioned, you can have many more infections if you start producing these spores in the orchard, and they can infect fruit further. So symptoms, this is something that we are used to looking, of course, in the Northeast and even in the big part of the Midwest, pretty much the mid-Atlantic in general. But one thing that, we don't talk much about is the glomerella leaf spot. We're starting to move towards looking at the leaf stage of this disease. These are angular spots, brown. They start as purple small spots, and they enlarge and then they become brown, and they have concentric rings in them. I've only seen this once in New York and Warwick at the south, but my growers in Virginia are starting to have more issues with it. Whereas this is the biggest issue right now, the leaf drop in North Carolina. So with more heat, you might, of course, and wetness, you might see more issues with the defoliation from glomerella leaf spot. One thing that I mentioned before with the fruit, those are the symptoms that basically looked like typical bitter rot. And when you come to North Carolina, where Sarah is, you see symptoms like this. And we can debate many hours what these spots are made of, but she consistently isolates Cholectotrechum from these. But these are not your typical spots that you would see. Yes, they would come later. She would have first defoliation, then symptoms on the fruit. But when you have a fruit like this, you can't with certainty say that you have bitter rot until you do the isolation and some gene sequencing. So there is this notion of course, not being certain which rot is caused by which pathogen. And there's other, rots as well. So what we have done when we came to Virginia to follow up what we have done in collaboration with Philip and Carrie is actually we went to locations and measured what the losses are. It was a big deal in 2022 and 2021, and we've continued rating to see how much loss the growers actually have in their farms. And it can be anywhere between, as you can see here, 20, 28, 30% up until, about 90%. So, and we have seen organic orchards that can lose more. We have seen actually cider apple orchards in Virginia here losing up to 98%, sometimes because just the sprayer broke down or the tractor broke down. They had to do service. They didn't have another to replace it, and the whole orchard was infected. So it can spill over in the post-harvest as well, and the initial reports were anywhere between 2% in New York up until 15%. So it can be a post-harvest issue too. This is the region that we covered mostly in the past. And these are the two complexes that Philip mentioned. The Acutatum complex above in the blue, it has temperature optimum around 77 Fahrenheit, and then the bunch of other species in Gliosporides complex that actually love temperatures around 86 Fahrenheit. So the ones in the yellow were the ones that were described in the literature throughout the whole world that were reported causing bitter rot on apples. Then, we have done the work in New York, and you can see about 50% is the split between the two complexes. The pink that you see on the right side of the pie chart is the Cholectotrechum furinae, and then the blue, pale and dark is the Cholectotrechum chrysophyllum is the dark, and the Noveboracensis species we described is the other cause of the disease in a small percentage, about 12% in New York. But then when we moved to Pennsylvania, the situation changed in the favor of more furinae. Of course, Gliosporides species were there, but there's more diversity. But when we moved to Virginia, we saw a different split. We saw that furinae is minority, and then the Gliosporides species, anything from green to to more intense pink, to nymphaeid, actually to the blue, those are the Glomerella species. So as you probably go south in latitude, you see more dominance of the species that are adapted to warmer temperatures. So it changes not only in relation to latitude, but also in relation to the relief or the altitude. So I would assume, this is my guess, that the reason why Philip sees the predominance, so it's basically 2/3 of Furiniae, is because of the more mountains they have, of Appalachian mountains. And then when we come down to Virginia, we do still see Furiniae, but the predominance is of the Glomerella complex. So we are definitely warmer. So Fatema, did some studies in my lab. She was my postdoc. And one of the things that we wanted to see is what is really different between these species. And I just want to make sure that you keep in mind that this is not the same thing as you have apple scab fungus venturia. You spray fungicides and you're done, and it works. This is a complex of different species, and this is proving that they do have different pathogenicity, which indicates to us, and we are pursuing this further, that they do have different genetic elements that allows them to be differently virulent. So you can see that some of the species are less virulent, some of them are more aggressive, like Siamensi. But it's very interesting. The Siamensi is the least present, whereas the Chrysophyllum in particular are highly present. So it seems that species that are highly dominant are less frequent. And then, I'm sorry, the species that are highly virulent are less frequent. And then the species that are predominant geographically, they have lower pathogenicity or lower virulence. So we started doing the experiments at Hudson Valley Lab, where Scott is now. And the first thing we wanted to do is really push out any interference from the spray programs the growers use and actually test each compound throughout the whole season to see how this works. This was published in Philip's paper. We contributed that part. We wanted to apply as what Scott mentioned. We would just use either Captain in treatment 2 throughout the whole season, or Omega at 2 different rates, 2 different treatments. And these SDHIs that you see from number 5 to number 10. And then we had one DMI Sevia, and we applied them 5 to 6 times throughout the season. You can see the spray dates up there on the right. And we had 3 different cultivars. And then we also inoculated, right after we applied our first treatment. And what we found was that out of all these materials, and when we look at the Honeycrisp that is the most susceptible here, you can see that overall, the only material that is from the STHI group is the number 7, the Aprovia, that was effective. And the reason we really put a lot of chips into testing all these materials in the STHI group, because it's the newest class of fungicides, but only Aprovia was effective. This aligns so closely to what Carrie found. Then you can see the Captain is doing a really good job, and Omega is a different other group, the FRAC 29, that did very good, at a higher rate. So then the Aprovia now and Omega are the new fungicides that potentially could be used during the summer to control bitter rot. And then they were also comparable to the Flint Extra, the most effective group of fungicides, the group 11, Ostrabilurins. They are the most effective, except Zoeren. So Flint Extra, Luna Sensation, Maravon and Pristine are very effective. And the slightly less effective is Zoeren. It's still effective statistically, but it's not as great. And I'll show you the data later. And then you can see the grower standard is number 14 here. This was the mixture of different stuff that you apply from every 2 weeks to 10 days up until the harvest. So this was the first thing that we did, and we did not have a chance to actually repeat this because of the pandemic and different reasons. And then when I moved to Virginia, the first thing that growers told me here was, we're concerned about customers coming to us and saying, do you spray pesticides? And if they say yes, they just leave the farm. So they said, we are interested in Central Virginia with new alternative materials that are more soft. So then the first group of the green materials you see here from number 1 to 8, it was anything that I had my hands on before for different other purposes, like fire blight or scab or sooty and fly speck, that I had my access to, and wanted to test it against the bitter rot here in Virginia, and then wanted to compare it to 9 to 17, which would be the single site synthetic fungicides that I've had experience with and wanted to repeat whether they're going to work, wanted to see whether they're going to work as well as they did in New York. And the treatment number 18 is the only one that is different, right? So that one you change every 2 weeks, you change the mixture or materials, whereas the other ones are all single material applied at the same times. So throughout the whole summer. And the guiding rule for us was either, applying the cover at 14 days or 2 inches of rain, whichever comes first. So again, the same concept, but now including the softer materials. And what you see here on the left side of the graph, the results from the fruit on Hydrate and Golden Delicious show that the materials that are softer or the more environmentally friendly, they're just not able to hold the fort against bitter rot, except statistically only is the treatment number 1, which was Regalia at 64 fluid ounces. And it's kind of counterintuitive because we see worse effect with the Regalia at 120 fluid ounces, which is the treatment number 2. We didn't expect that. But it seems that lower rate works slightly better. But again, the grower in this case would withstand about 38% fruit loss, which is not acceptable. And then the synthetic materials are all performing really well, except the black sheep in the whole group of Frac 11, which is number 10, and that's Zoeren. It just did not work well. We don't know whether this is because of the specific mode of the action of this compound, or maybe there's resistance to it. I really don't know. We didn't look at that. In my trials, what you can see here, I always use 2 different controls. I use the inoculated control, untreated inoculated control, and also the untreated non-inoculated control. So I can see what the pressure naturally is from the bitter rot here. And we do have a strong pressure, as you can see in this block. This is a classical block. And so we also rated this disease again in 2023. This was a drier year. We had a really wet year in 2022, but this was a dry year. And again, you can see by and large, the treatments that are soft from 1 to 8, that are environmentally friendly, they're just not able to hold the fort against the disease, especially on Hydrate, that is far much more hammered by bitter rot in comparison to Golden Delicious. And you can see here that we did get better results in this case with 64 fluid ounces of Regalia treatment number 1. And of course, it's performed better because of the better weather conditions. That's what we think. But we would probably not recommend it, even though it's statistically different from the control, because it's just not creating as good of a control as we would hope for. So the trick would be potentially we might incorporate it into a program, but maybe in the drier times. But it will be nestled in between synthetic materials that you apply before or after something like this. Again, you can see here that materials that are synthetic from number 9 to 18, they're all worked really well. You can see that the Zoeren performed even better because it had drier year in which it does show more disease, but all the other ones were really working well. And one thing that I want to mention here, I didn't mention in the previous slide, you should pay attention to Xyrem number 15, 16, and 17, and Captain and Furbam. So right now there's the PID on them from USDA. They're being reevaluated whether they're going to stick around or not in apples. We don't know, but they are the backbone of the resistance management. Meaning, if you apply single site fungicides like the ones that we have here from 9 up until 14, if you apply single sites, you should always tank mix them with the contact fungicide so you can broaden the spectrum of efficacy of your spray mix, and that way you prevent potential resistance rise for the single site fungicides. So if we lose them, it's going to be really concerning. How are we going to do that, in the future? So right now, what I'm recommending to my growers is that they should not rely on any soft materials because they're not effective against the bitter rot. So these synthetic materials did have a control anywhere between 80 to 100%. Okay, and again, Furbam, Xyrem, Cap10, Omega, and Aprovia were really good, reliable tools. You could definitely use the contact materials with the Group 11 fungicides, and the Group 11 should be alternated with Omega and Aprovia because they're different groups. 29 is Omega, Frac 29, and Aprovia is the only one in the Frac 7 group that is effective. So you should alternate them throughout the whole summer so that you don't rely only on the Group 11. And when you incorporate these materials into the spray program, it might be more expensive, but it's going to pay the dividend down the road because what we don't want to lose is the efficacy of the Group 11 fungicides, which is your Mervon, Pristine, Luna Sensation, and Flint Extra. So you have to incorporate omega and aprovia in it, because if you apply these, and we have seen growers apply still these materials for during the season up to 8 times, you're risking potential resistance in Colotartrium species to this group of fungicides. So the biorational materials had efficacy anywhere between 25 to 75%. We did get about 60% control with Eugalia lower rate, but we can't really recommend that to the growers. So then we followed up in 2023. We wanted to expand more because we had another part of the block we could use, because we stopped doing the other treatments that I showed you early. So we wanted to expand more into the green materials, or the environmentally friendly biorationals, we call them. So you can see here from 1 to 4 and 7, these are the things that I wanted to put my hands on and really test them, because the previous literature was saying from Alan Biggs and Dave Rosenberger that calcium can contribute to the control of the disease. So that's number 7. So we had this formulation, Brexil calcium, in the shed. We wanted to try it, and then we wanted to go into more biocontrols. So Thea and Howler are the ones we tried. And then we also looked at some other fertilizers like Nutrify, Magnum, and Kendall. And then we wanted to implement more synthetics like Switch and Intuity, so that we can see how fludeoxonil, which Philip earlier mentioned as a post-harvest material scholar, wanted to see if it's going to do anything in the field in this formulation with cyprodenol. And then again, we included aprovia, but now higher rate. Instead of 5.5 ounces, I used 7 fluid ounces. And wanted to see how this worked. Again, same concept, one active ingredient applied as a spray program up until the end. And what we found was that, again, we see largely good efficacy to poor efficacy of the synthetic materials. Like you can see that Intuity, which is in the group 11, FRAC 11 fungicide, that's a newer generation, mendestrobin. It does a good job, but it still allows about 30% of the disease. Switch was about the same, and then the aprovia was the best out of all of them. But then when you look at these, fertilizers and the biocontrol, they do reduce the disease, but they're not, commercially acceptable. So you can get, control about like 30-40%. They do statistically reduce the disease, but they're not as good. We do see that Brexil calcium has done something too, and Kendall out of all of them was numerically the lowest. And so these synthetic materials, gave about 50 to 90% control, when there's the other materials were not as great. So we repeated this trial, of course, also in 2024. So this is, we're planning on publishing this by the end of this year. And you can see here again that the treatments start to fall apart more in a year that was dry. We had a very dry year, 2024. Largely we cannot say that you can rely on these fertilizers and biocontrol agents. They're not effective. In this case, we can see that number 13, which is Thea, and number 7, Howler, they completely failed. And in this specific case, I'm not sure why the numbers here are different from what I'm seeing here. They should be numbered differently. Something got garbled down in the labels, but they should be on the same spot at least. You can see here that Switch and Intuity did do a good job, but aprovia still was the best. So the 6, the 2 here, the blue, and the aprovia were good as the grower standard. And so anyhow, we have 2 years of data to show this. This stuff is just all around the biocontrols that we have currently. They're not effective in the mode of application that we have right now, and this is like every 14 days or 2 inches of rain. So the company approached me and wanted me to test whether we can reduce the spray interval, from 14 to 7 days. So we did do that. And so, just to summarize this section before I go into the next, again, synthetic materials are really the kings. The group 11 here in blue are really effective. Aprovia is the next, omega is the next one. It is good. And then you have to mix them all whenever you apply them with these multi-site materials, so that you can make sure there's no resistance in the future to the group 11. And some people are just concerned about the price of implementing aprovia and omega, but I think it's very worth the money. Biorational materials, again, they had high percentage of bitter rot. They allowed between 25 to 75% of bitter rot. And in general, we see some statistically significant effects, 30 to 60% control, but they're probably not going to be acceptable to the growers. So if I, in an ideal world, would want it to see growers do textbook example spray programs, this is the way how I would envision one for the Virginia and New York. And the reason why I would like to see this is because, first of all, I start the season with the first cover, which is about 10 days after petal fall, with materials like Inspire Super and Topsin and Profite. Mind you that these are not effective against bitter rot. I still apply them early on up until the third cover because there's still more issues at that time, probably with scab and also with powdery mildew and cedar apple rust. They're extremely damaging, so you still have to continue spraying for those. And the pressure for us goes up for powdery mildew extends up until the third cover. So we still are concerned about that. And Captain probably is picking up any slack here for bitter rot control if there is any early buildup of infections. But by the time when we believe that the infection pressure becomes really high, which would be a fourth cover, I would start applying the materials such as Aprovia with Cap10. Then I would switch to Flint Extra, and you can do 2 Aprovias back to back as long as you don't do the third one. So now you should switch to Flint Extra and then do the Omega and then finish the season. This is the way how I would do it, finish the season with materials that are group 11, the most effective ones, especially using the ones like Maribon that have 0 days pre-harvest interval before just the harvest. Later maturing varieties, of course, would have to be, sprayed all the way up until harvest, in November maybe, because some of these latest varieties, they just get forgotten. They get bitter rot because the growers become busy picking and they just leave them unsprayed, but they do have to be sprayed all the way up until the optimal harvest date. So you can have larger number of these cover sprays, depending if the variety is late maturing or less of these cover sprays here, maybe about 7 to 8 in the early maturing varieties. So then what we did is with my new students coming in, we wanted to, of course, to play more with different materials. These are the problems that we are facing that I mentioned to you before. And also, we are concerned with spraying multiple different species with these materials. So it's not only one pathogen, there's many different ones in there. So they might have different strategies in the way how they acquire resistance. So and right now the EPA is reevaluating these materials. And one of the things that we wanted to look really was expanding into synthetic world. So Quinoprol is the material that is in the it's called ipflufenaquin. It's a new synthetic material. Just somehow my mind is escaping right now what the group is of ipflufenaquin, but it's going to come up soon. Then we wanted to test something that we heard from Anissa Palatovich from UNH, that if she got good results with Carrie Peter with tidal growth spectra. We wanted to test that one. That is definitely a birational material, made of it's made of shells of lobsters, I think. And we wanted to go on and follow up on howler and thean and reduce the spray interval. So looking more into the biocontrols. So these are the active ingredients that you can see here. So the reason is a group 11 fungicide. Okay, so it is. Then we followed up on Kenja, a new group 7 material, and then we included the Luna, Topsin, and Quinoprol, because people always ask me whether Topsin has efficacy against bitter rot. So we'll see now. Quinoprol was the group 52, actually. It's a new group of fungicides, but the reason is the group 11. We heard some efficacy exists, and then we looked at these materials that are more birational, like sulfur and tidal growth spectra, and then included some biocontrols. So howler, thea, and a YSY yeast. It's all of them are biocontrols there, and then we had the grower standard with different fungicides alternated every 14 days. So we had 2 different controls again. And what you see here are the results again on Iterate and Golden Delicious. You can see that overall, the really good materials that did do something were the Reason, far left, then the Luna Sensation. All the ones that have a yellow highlighted letters above the bars are significantly lowering the disease in comparison to the control. And one surprising thing is that the tidal growth spectra was really effective. We're happy about that, and it was statistically similar to grower standard. But the sulfur and YSY yeast did not work well. But one of the other, surprising things is if you look at the far right, howler and thea were giving also good results. In this case, still all of them are allowed about 20% of the disease. Not really great. Okay, but keep in mind we are applying this one active ingredient throughout the whole summer, and I think that at that point the formulation of the material becomes a factor. And I hope maybe if we mix them up with other materials, they potentially could be as effective as in a spray program if we apply them in a mix or alternation with other materials. So we had the biocontrol giving about 50 to 53% control. Synthetics were really good, between 40 to 50% control, and biorationals, had some effect. They were the best in this trial, about 60% control. So we have these different things that we highlighted here, not really important. We also rated the disease about 7 days later. Okay. And the reason is once we start rating the disease, okay, we don't come back and reapply these materials. We kind of wait for about 7 days, and we want to rate again so that we can show you how quickly these materials fall apart. So, and of course, we were due at the time when we rated on July 22nd, so it's about, 15th to 22nd, 7 days. Things start to fall apart really quickly. And you can see here that even with synthetic materials, it did not work well. But number 9, tidal growth spectra, and number 17 did hold the fort further. So this is where we start talking about the residual of the materials. And we also see that howler, number 22, did do numerically well, but it was not statistically different from the control. So we just show this so that you can actually, keep in mind that these do require reapplication all the way up until the harvest, because they can quickly fall apart and allow infection to occur. Okay, so we rated in this trial also Glomerella because it's a concerning thing for the growers here, and the results overall were just less intuitive. Okay, by and large. And the reason why is because we are in the northern part of Virginia, where Glomerella is not really a great problem. But when you come to central Virginia, that's where the growers have issues with it. But in general, you can see number 3, Kenja, again, highlighted letters in yellow indicates to you the bars that are effective. We see YSY yeast now being effective, and we see the grower standard being similar to them. Pretty much largely all of the other ones did not perform really well, including the biocontrol, which was the worst. But the biorationals were about 57% effective, and synthetics anywhere between 40 to 50%. So this is just the first time we started to work with this. We really are getting our feet wet here, and hopefully in the future we're going to get better results. So this was the rating on the spur leaves that develop early in the season. So keep in mind the biology difference. But then these are the shoot leaves. So you can see here we have much better results on iDRED. Overall, you can see the purple bars show a good significant effect of anything from 1, well, not maybe 1 to 4, like 4 is not, but anything to 1, 3, 5, 8, everything there that is in yellow is good control in comparison to the untreated control. But in general, we don't see really good effects on Golden Delicious. So it seems that in this specific case, the variety might be another factor. But you can see about 70% of synthetic control, or up to 80 in certain cases, about 77% control with the biorationals. This is the first time we're doing this, so we're hopefully going to learn more as we do it in the future. So finally, just to wrap up, it seems that we do have efficacy of the biocontrols, but it's anywhere around 50 to 55%. You have to apply it frequently on a 7-day schedule. It's better. Tidal growth spectra seems to be, based on 1 year of data, working really well. So I would potentially see it in the future as a part of the spray program that growers would use. So we have to repeat this next year so that we can actually see whether this is going to really work again. And then we did see some good control with the glomerular leaf spot, but in general, the pressure from the disease was low. So we can't really, make conclusions count that well. So we're going to have to do more testing. Before I finish, this is, one of the last slides before the last one. Is that this is the recommendation that I've heard from Dave Rosenberger and kept on parroting it, because I've had a lot of growers who approach me and ask, I have bitter rot right now, but what do I do right now? Should I spray or not? Should I just stop and waste the whole orchard or not? Dave's recommendation was always that you should definitely apply full rate of Captan with Pristine or Maravon and add some LI700 to it, because there still might be fruit in there in the orchard, a certain percentage of fruit that are not infected yet. So, and then repeat it, again, after 10, 14 to 14 days. So you might still be able to have some profit from the orchard. You should not really let the whole orchard go. And the reason why is because then the bitter rot pressure is going to increase. So you're going to have an issue of higher inoculum next year. So you should maintain the coverage and, reduce the stress in the orchard. I completely agree with Philip that there is indications that the orchards that are drought stressed have more bitter rot. And also you can have more bitter rot overwintering apparently on the fire blight strikes. So the good sanitation is not only applicable for bitter rot, but it should be also done on other diseases, because that tissue can be inhabited by the bitter rot. So, and finally, just want to wrap up with this, that, this is really it takes a village to do all this. It's not easy, and all the funding sources that allow this to happen. So I'd be happy to answer any questions that you might have.
Great, thank you so much. Yeah, so we have about 10 minutes for questions if anybody has any. If I'm gonna be quiet for a minute to let anybody jump in before I start asking my question. Or if nobody else has any, I was wondering about when you were talking about the different management options. So bitter rot, is present in Western New York, but I would say probably not at all in the same scale that you guys are seeing. I'm wondering, in terms of all of those cover sprays that your growers or that you're recommending, what percentage of those would you say are sort of specifically targeting bitter rot versus what percentage are targeting, something else, and then bitter rot is sort of coming along for the ride? And then kind of the follow-up to that would be, how does that change as we move away from the Zoeren and the Captan and move more into the biorational or the biopesticides?
Yeah, that's a good question. For us in Virginia here, the bitter rot basically became the top problem during the summer. So we have these early, spring diseases, apple scab, powdery mildew, and, cedar apple rust. They're really, really damaging. By the time, when about mid-May comes, we switch to think about bitter rot. And in the past, it would be mostly stuff that they were concerned would be black rot, white rot, and pseudobacterium fly speck. But it's shifted in the last 15 years from those being something minor that shows up, irregularly, whereas the bitter rot is showing up almost every year. But one thing that growers tell me is it is there. We see it every year, but in dry years, we can control it easily. But when we get wet years, like 2022 for us was extremely wet, they see stuff that they apply regularly fall apart. Just there's gaps or from rain. So for them, during the last, I would probably say 10 years, the bitter rot became the primary reason why they spray, whereas in the past it would be black rot, white rot, maybe stem end rot. And I can be corrected by Bill McIntosh if he ever hears anything you guys reproduce from me. He might have better knowledge of maybe what would be in the past in terms of what they really were concerned about. And then what was the sub question? You said something about
I was just wondering if that sort of the guiding principle of those sprays would change as we move into the biorationals or the biopesticides, if you're having to maybe double up sprays in order to get both.
So if we have now an issue of not having the Zoeren, right, or maybe limited to Captan. I think it's going to become very difficult. I see Christy Grigg McGuffin is here. They have already lived through a certain number of years right now from not having these materials applied, because they have it's basically the reason what has driven the heavy limitation of Captan in Canada. It's probably going to happen to us too, is the labor exposure to the materials, to Captan specifically. And I think, and then, cancer related diseases that occur after that, so right now I think there's the limitation. Once you apply it, there's a certain number of days, and it's pretty excessive, certain number of days that you cannot go into the orchard to work. So yes, I think we are desperately right now here reeling, to try to find anything that can replace the Zoeren, because there's a lot of growers from all the way from Washington down to Georgia that love Zoeren. It's cheap, it's effective, and we most likely are not going to have it anymore. We did definitely lose Tyrem, and I'm concerned about the future limitations of Cap10. Furbem is completely gone out of the picture as well. So we're trying to find something that is multi-site, and I'm hoping Tidal Growth Spectra is going to maybe fit the bill in the future. But I think it's going to become much more concerning for us from the point of view of managing resistance, specifically the fungicide resistance in the Colotartricum species. That's where we get concerned. Now if you start becoming dependent fully on the single site materials and only maybe you have one or two multi-site materials, how sustainable is that going to be in the long run? Because we do tell growers apply only in the whole season, you can only apply 4 fungicides in the group 11. So then we get issues with people saying, well, I'm still using it for powdery mildew. It's one of my standalone things that I always do with pink, and I do it at petal fall. And I said, well, move away from that. Don't use it there. Use, DMIs, use STHIs for powdery mildew, use sulfur if you need them early. And just forget about powdery mildew. Save your 4 shots of group 11 fungicides for summer. And then, people come still to me and they say, well, the label does not limit me to apply more than 4. And yes, some of the labels are vague. Some of the group 11 fungicides. And, they think that the label says, yeah, I can apply 4 Flint extras, 4 Marivons, 4 Pristines, 4 Luna Sensations. No, that's not what you should do. Absolutely not. That's, even the label may be vague. Some labels might be vague. That's not what you do, because all of them are in the same group, and you should limit yourself to only 4 applications. We are trying to push them to use Aprovia and use Omega as alternating, materials, and then also tank mixed with these multi-site materials. So yeah, that's what all I have as an answer to that one.
Srdjan, if I can just jump in, I'm
Sure.
curious. As you mentioned, we're dealing now in Canada with our loss of group M's. So from a tank mix perspective, are you starting to think of alternatives then to Captain? Because for some of our larger orchards, we're only allowed 2 applications. So what's our alternative then for a tank mix?
Well, the only thing that I would do would be to have 2 single site materials mixed together. Okay. And I don't know what I would do, maybe do Aprovia together with Omega, or I would do, I don't know, Luna Sensation together with Aprovia. I would do something like that. Luna Sensation already has something like some other SDHI. But I'm hoping if you guys have Tidal Growth Spectra, if it shows to be consistently effective, I'm going to start doing more experiments with, tank mix of Tidal Growth Spectra with. I don't expect it to be ineffective when you tank mix it with single site fungicides. I think it's going to be, it's not a biocontrol that maybe could be affected by a fungicide. I think it should be effective, but that's what I'm hoping for, that we should start using Tidal Growth Spectra as a replacement for these materials. I wish we had more of those, as options, but I haven't heard. If anybody has something to suggest we should test, by God, send it to me.
Have you done any work with Buran? That's the garlic extract.
I did do Buran, and I tested it in New York against Sudobotchin fly speck. Okay, and we applied it alone and with oil, and it seemed that it did do a good effect in reducing the severity of Sudobotchin fly speck. So you would have less spots per fruit, but you would still have some spots. I did not, Claude, the guy who's in the company, it's a little bit tough for them to send these big batches. I haven't tested it for bitter rot. I would love to test it for bitter rot, but from what I've seen with Sudobotchin fly speck, I'm hoping maybe it's going to have some effect. I really don't know, but it's just difficult sometimes to get the material down here from Canada. But hey, if we can get the batch to test it, it will be great.
That's great. Thanks. Yeah, we're struggling with trying to how to place that because from a scab perspective, then it's more of a post-infection application. So the idea of then using it as a tank mix, it gets complicated then come summertime. But yeah, appreciate that. Thank you.
Yeah, I mean, I really feel for you guys there. I really don't know how and what you do, but I've heard, maybe I should not say this, but some growers told me like, we still use it, but we don't tell anybody.
Because people have gotten creative.
Exactly. But then again, I'm wondering when, an example arises of like, oh, we found this happen, or somebody, EPA comes in and catches somebody, or maybe USDA catches somebody, inspector comes in, right? We have pretty strict, pesticide inspectors in Virginia, and they can catch you doing things wrongly, and then you can pay a huge fine. In some cases, the fines can be so high that you could, shut down the whole farm. So what I'm trying to say is, yeah, I think that it's a big problem for us right now, how to replace stuff. One guiding principle of the USDA and EPA is to use science to make decisions. Right now, as far as I have heard, the toxicology tests for different groups of animals have changed in the last 10 years. I really don't know the nitty-gritty details of the whole thing, but the toxicity and sublethal toxicity they're measuring right now, that's also one other underlying factor why these materials are being reevaluated.
That's great. Thank you so much. Thanks to both of our speakers. It is just about 1:30, so I think I'm going to wrap up unless anybody has a really quick question they want to chuck in right now. But if not, you guys, I think you all know how to get a hold of both Srdjan and Philip. If not, you can always ask and we can provide their contact information for you. Really appreciate you guys both talking with us, and it is absolutely a really big problem, something that we're all looking at and watching and looking for recommendations going forward. So I appreciate all the work you're doing on that. Thank you everybody for attending today. We did record this session, so this recording will be made available. I'll send out an email to the full group for anybody who wasn't able to be here or who wants to rewatch a portion of it. And then again, we do have 3 more of these sessions coming up. The link is the same for all of them, so I'll send a reminder email. But you can just go back to the same email and click that link next week. Our next one will be also on Thursday, a week from today, and we'll be talking about these updated EPA regulations. Of brief review of the FIFRA program, discuss recent changes, Endangered Species Act, worker protection standards, all of these things that we're all thinking about way more than we want to. And then again, there will be 2 more the following Thursday, and then we're going to skip a week, and then the final one will be on the 13th. But we'll remind you about all of these by email and in the next webinars. So if anybody has any final questions, go ahead. But yeah, thanks everybody for joining us, and I hope to see you all next week.
Thank you.